Yeah, my name is Emmett Shearer, Senior Manager of Clinical Education for Therapeutics, a small emerging company. We'll talk a little bit about the products in the company towards the end. But we're going to talk about bone grafting today. What's interesting is there are well over 300 bone grafts available for use throughout the industry in the world, especially in the United States today. But it's going to be kind of curious as to how many of them even have any real evidence that they work. So the agenda is we're going to take a look at the options, and evidence is one thing we're going to focus on fairly heavily today. Look at autograft, allograft, EBM, BMP, synthetics, cellular-based products, I think commonly referred to as stem cells. We'll talk a little bit about peptide-enhanced bone at the end of it here, and then we'll take a look at the evidence that's actually out there. There are a couple of ways, or three different ways that actually bone graft materials can make it to the market. If you look at the top two there, these are products that have gone through a PMA IDE study with level 1 data with FDA indications for specific applications. This is like a drug-device combination product makes it to market. There's also a 510K product system to get to the marketplace, which is you have to be able to demonstrate that your product works equivalent to another product that's already on the market. Synthetics and DBMs come through the marketplace and make it to the market that way. Also, HCTP products, human cellular tissue products, is another pathway, and this is how allograft chips, structural allograft, make it to market. And remarkably as well, the cellular-based products, when these come through, they don't have to demonstrate safety or efficacy, just that they're processed appropriately and brought to market. So it's interesting that there really isn't a whole lot of data out there to support their use at this point. And again, here's just a little representation of what levels of evidence really are out there, what kinds of products have to come through those specific pathways, and really how there are several different thresholds and levels to be able to make it to the marketplace. Really, ultimately, what you would like to have is prospective human clinical data that actually supports the use of a particular product. The marketplace today has varied quite a bit, and we have everything from the BMPs and growth factors to allograft to BMP to DBMs and to synthetics and so forth. So the marketplace is really quite well divided as far as what's being utilized. But again, the data is, I think, the one thing we have to take a look at. Just to categorically look at some of the options that are out there, Autograft, of course, is the gold standard. Even the FDA today, whenever you're going to bring a product to market, does say you have to use Autograft as a predicate device. We know how it works. There's a lot of evidence out there. Historically, it's worked incredibly well. Actually, nothing has been proven to be clinically superior to Autograft at this point in any prospective trials, and that's why it still is considered the gold standard. It happens to have what we need: the scaffold, the cells, and the signals, the three properties for bone formation. And that's why when you harvest it and from usually the iliac crest and transplant it, it performs quite well. Sometimes we don't have enough autograft, or it's difficult to be able to get the autograft for the patient. Allograft was, I think, the very first of the other options that were available, and it comes in a number of forms: allograft chips and so forth for augmenting, say, a spinal fusion. There's also structural allograft as well for fractures and so forth. In the literature, its advantages are it's fairly inexpensive, and in low-demand applications and good applications, it actually has been demonstrated to be efficacious. But some potential disadvantages are that there's always going to be the potential for the transfer of infectious agents. Sourcing has been an issue over the last several years. I think they're getting a little bit better handle on that right now. And again, it's only comes through the FDA as a HCTP product, so they really don't have any regulatory authority over the use of it at this point. DBMs, the demineralized bone matrix, was actually handled the same way for many years, but a few years back they decided to have these companies at least produce a 510K comparative animal model to demonstrate that it'll actually work as well as those that are already on the market. So this actually comes through as a 510K. DBM essentially is bone that's been demineralized with hydrochloric acid to actually release the proteins. Of course, the major proteins in the body are collagen. Associated with collagen are also some non-structural proteins, and they include some of the TGF-beta, TGF-alpha factors, and especially when we think of this BMPs. It was Dr. Marshall Urist who was studying bone. He understood that cells do all the work, but how do they know where to go, what to do, how to multiply, and so forth? And he was one who postulated that perhaps there's some growth factors associated with these proteins as well. So he demineralized the bone matrix and implanted it, and actually in mice initially, and found that he could induce bone formation. So from then on, industry began looking very heavily at osteoinduction. We'll talk about that as we get to the BMP section as well. One thing that was demonstrated over the years is that these lots of the DBM products have an incredibly variable amount of the BMPs associated with them. This particular one, just look at the top, it doesn't matter what the product is, you can see that, bless you, from 97.5% to 28.2% as far as the BMP concentration of BMP2 in this particular offering. That's quite a large coefficient of variability when you're considering it. Which one are you actually getting when you pull it off the shelf? The one with 28.2, the one with 97.5. We really don't know, and that's why it's been confounding to a number of investigators over the years. They've not been able to really replicate any study where they've had a specific result and have it quantifiably replicate exactly the same with the same product because of this high level of variability. Another thing that's interesting is these are measured picograms per milligram. At the fracture site or within a marrow space in humans, BMPs are expressed in nanograms per milligram, so it's about 1,000 times greater in concentration. So while this was a really good experimental tool that Dr. Marshall Uris utilized to understand how to induce cells to become a mesenchymal stem cell to become a bone-forming cell, proliferate and form bone, was excellent. But he was not an advocate of utilizing DBMs clinically in human beings per se. So he wanted to utilize it to find out what is the best factor we could find and how do I purify it and how do I bring it to market. A number of other studies have demonstrated that the DBMs as a standalone product tend not to work nearly as well as Autograft, and we've had some variable results with those both in the preclinical and clinical series. There have been a number of studies done up from level 1 to level 3. The one takeaway from all the studies is that DBMs can increase the handling of what local bone is available, perhaps. They can also help extend Autograft as you're applying it. And so as an extender, they do have some merit, but as a standalone product, they've never been able to perform anywhere near the efficacy level that Autograft can perform at. Well, since we're talking about osteoinductivity, we'll talk about something that really does work. BMP, bone morphogenic protein, it's recombinant in human. BMP number 2 is what Infuse was approved as. It's a product that had to come through the FDA with the PMA IDE study, and we'll talk a little bit about the structure of the study in a few minutes. This is Dr. Marshall Uris. He actually wrote the paper, Bone Formation by Autoinduction, 1965, and he was being considered for the Nobel Prize because of this work. So it was an incredibly seminal and important work at that time. It also took us into and helped develop a whole brand new area of molecular biology, because he was one who was saying, hey, we have all these different types of growth factors that are associated not just with bone marrow and bone, but all over the body. So his seminal work was really important throughout the entire course of medicine today. As I said, he's the one who coined the term osteoinductive because it induces bone formation. And at this point, industry began looking very, very heavily at osteoinductive aspects. But also we have to have osteogenic and osteoconductive components to bone as well. BMPs are good, they work, there's no question. However, they're highly conserved proteins. The BMP in a Drosophila, the old fruit fly that we studied in genetics, is roughly 78% the same as ours. So, if you look at the phylogenetic activity of it, it's been around for a long, long time, and it doesn't just grow bone. It has activities in everything from embryonic development to tissue development to right side, left side body patterning to cephalid and caudal body patterning. It's a very important protein that turns on and off at several different times as we develop as human beings. So we should use it with caution, and I think that was what Dr. Edgar Dawson, who was Marshall Uris' partner, stressed in a letter to the Spine at one point when products first came to market, and I think people who heeded it did very well. There have been some published clinical concerns in the literature. You're all familiar with the ACDF applications with the inflammatory response and swelling and so forth. Alex Vaccaro noticed late radiculitis when he was studying it. Ectopic bone formation, osteoclastic hyperactivity have also been noted a number of times and written about. And as far as posterior lumbar fusions, it was always always an issue because of basically the carrier, and people have been working on how they can make it work better there as well. But these are some of the actual published concerns that have occurred. The FDA study itself was a prospective single-level study. It was 288 patients, 139 controls. They actually had the outcomes of CT, oswestry, neurologic, and so forth. The fusion success rate was 93% versus 88% in controls at 2 years. But when we actually look at the combined or composite overall success rates, it looks like it says 57.1 versus 56.7. So it was considered statistically equivalent to Autograft. Its main claim, though, is it obviated the need to go to the iliac crest, relieving the pain and morbidity associated with a second operative site. But it works incredibly well. Anybody who says it doesn't understand the product. We just have to be careful how we use it. And I think some of the issues really have been more of a carrier issue more than the product itself, actually forming bone. Synthetics. There are 185 different synthetics available, I think, on the market at this point. And they range from a number of different chemistries. Most important aspect about a synthetic product is what's the chemistry and what's the structure. The chemistries are everything from calcium sulfate to hydroxyapatite. Calcium sulfate, of course, will dissolve when put into an aqueous medium. So could it be used as, say, a carrier for some type of an antibiotic? I think people have mentioned that in the past. Whereas hydroxyapatite, at least in the synthetic form, is in a slightly different mineral phase than the hydroxyapatite in our own bones. So it tends to take a long time, if it does even resorb over time at all. Some studies have demonstrated that the persistence of that material in there for up to 4 or 5 years post-implantation. People have tried adding collagen to it or making a biphasic type of a TCP product or a calcium phosphate product with some degree of success. There are only about 3 or 4 that have any level 1 to level 3 evidence, and usually they have to be mixed with bone marrow because that brings in the cells and the signals to physiologic concentration. But again, there's so many options out there today, and most of them only have basic animal data to support their use. We hear the term stem cells thrown around quite a bit. Within this purview, we're looking at stem cells we considered allogeneic stem cell systems. Essentially, all the major spine companies out there have some type of an offering in this area right now. The only one that doesn't, of course, is Medtronic. Of course, they have the BMP, so they don't really need it. The FDA approval, as I said, regulates it as a human tissue, so it doesn't require any kind of FDA approval for safety or efficacy. And the sources, of course, are donor bone, and where does that bone come from? We don't exactly know where it all comes from. However, there are some corporations, MTF, for example, that has a very good screening and processing methods. If we look at the in vivo data preclinically, this is just some original animal data. This particular design was a posterolateral fusion study, and they were looking at this product called Trinity. It's just one of the products that are out there. And they looked at it versus Grafton. They looked at radiography at 2, 4, and 8 weeks in the animals. They looked for palpation as well as micro CT, and they actually found that there was no statistical difference in bone formation, so there's really no advantage to adding mesenchymal stem cells, as they call them, the allogeneic stem cells, to the mixture versus having Grafton or DBM by itself. So this was one of the first independent studies that was looking at, do these things really say what these people are trying to claim they do? There are a few studies available, but they're all very, very small in the end. Some are as small as 8 to 13 patients. Some are as high as maybe 40 or 50 patients. Most of them are open label studies, so there's not a randomized control aspect to them. Just to compare a couple of studies here, one of them, of course, was a company-sponsored study. You can see on the left, it was an ACDF, 31 patients, and they looked at 12-month radiographic fusion success and 12-month changes in NDI and VAS. And they found that they had a 93% fusion rate and improvement in the VAS. However, if we take a look at an independent study that was actually done in spine, not supported by any of the corporations that are associated with these. Again, it was an ACDF perspective, but they had a randomized control that was retrospectively given to it. 57 patients, and they looked at 12 months. and they found 87.7% fusion rate in the osteocell. On the other side, they found that a 94% fusion rate in the allograft only. Their conclusion basically was that there was no statistical difference between the two. Therefore, is there an additive advantage for having the high cost of, say, an allogeneic stem cell? Again, they found that it really wasn't a huge improvement over what allograft alone can do. The FDA is also taking a look at this. They have other stem cell products that are on the market that have gone through FDA approvals, and those are actually listed as drugs. They have to go through a PMA IDE study. And they're looking now, over the next 3-year period or so, with their guidance to the corporations that are selling these products in the marketplace to probably make them go through a PMA IDE to at least demonstrate safety and efficacy of the products going forward. We should hear a lot more in the next year to two from the FDA on how they want to proceed with this, but that's, I think, a good step forward. Again, it's human tissue. No real compelling human clinical evidence. We really don't even know if they're converting bone, because if they do, then the bone's coming from the donor, not necessarily the recipient, and it's going to express immune histochemicals and perhaps trigger an immune response. So that's an issue that has to be considered as well. They're also, basically set in the same price range as BMPs without really any comparative data. And handling has been variable with them. We're going to switch gears a little bit here and talk about a different product, or different, I should say, approach to bone grafting materials. It's a peptide. A peptide, of course, is part of this amino acid sequence is usually part of a protein. This new technology has been around since about the 1990s. We'll talk a little bit more as we go through here on the evolution of the product, but collagen, as you can see, is incredibly important. If we look at bone, it's basically of calcium phosphate and protein, but the majority of the protein, of course, is collagen. And collagen plays an incredibly vital role in our body at all times. It's a combination of 2 alpha 1 particles and 2 alpha 2 particles or 1 alpha 2 particle that actually wrap themselves up in a trihelical structure. And then, of course, they're able to bind into fibrils, and then the fibrils covalently bond into fibers, and the fibers go on to actually develop into tissues. And you can actually see this from the amino acids through the peptides, through the fibrils, the fiber, and ultimately into ligaments, tendons, and other components of skeletal structures. Bone itself, just as a basic look at it, is you have the periosteum, of course, which provides most of the nutrients and so forth to the bone. You have the osteons, which are made up of lamella, and we'll take a look at one of these. As you can see, we have the concentric lamella that are actually wrapped with collagen as well. This is where it actually gets it has compressive strength because of the stacking of the calcium phosphates, but it also has this bending strength because of the wrapping of the collagen. So if we take a look down a little bit further, the mineral structure of bone are these little hexagonal crystals that actually align and stack themselves up, and then they're wrapped with collagen. And those bundles are bundled together and are wrapped with collagen and so forth. So you have a very strong compressive strength as well as very good bending strength in bone itself. Collagen is also incredibly important for a number of different reasons. If we take a look at how collagen, type 1 collagen, is actually associated, you can see that it does look fairly similar to, say, the type of steel you would see in concrete. So it does really lend towards the nature of the collagen, of the material, to have both the bending and compressive strength. And gram for gram, it's actually stronger than steel for tensile strength. So it's really incredibly important for the body. Another thing that we found with collagen, or the researchers have over the years, is it also is very, very active. There are these things called collagen interactomes. And what they do is they actually have a huge influence on cellular interaction, especially attachment and so forth. We'll talk about that in a minute. They have ligand binding, and they also have over 1,000 different mutated diseases associated with collagen itself in a number of different organ and body systems. So as far as the interaction domains, P15 peptide actually falls within a domain that is very, very active for cellular attachment, and that's what the P15 peptide is known for. We found that it's actually something that is somewhat shielded by the body because it has such an incredibly high affinity for cellular attachment. So as we move forward on it, we found also that it's incredibly important evolutionarily. If we look at well, there was some research that was done looking at Tyrannosaurus rex versus a mastodon, and within the particular segment that they were looking at, you could actually notice that the P15 peptide and that epitome there were exactly the same between the two. So it's a highly conserved aspect of collagen as well. I mean, much like a BMP is so highly conserved. So it's very, very important. It does stimulate integrin interactions, as you can see. The peptide will actually stimulate and activate the integrin. The integrin then again begins to affect genomic activity in the cells, the expression of mRNA and proteins and so forth. We'll talk about a couple of those as we move forward. So if we take a look at peptide versus a protein, they're really quite different. Peptide is part of a protein, but it's not as large as a protein. It also, let's see, get back there. I'm sorry. There we go. It's also more specific in its activity, and it's less expensive to actually utilize. It doesn't have the same types of cellular interactions that a number of proteins, and especially the BMPs, would have. So they're both very efficient and effective products, but we decided to look at the peptide system and find out if we could actually produce the same amount of bone formation and so forth that we can say with a BMP, but without a number of the adverse events and side effects that were associated with it. There are a lot of osteobiologics out there, but P15 peptide has been demonstrated over time to actually have the strongest and most profound effect on attachment, differentiation, proliferation, matrix production, and bone formation. And that's one of the reasons it's being looked at quite heavily throughout the industry at this point. This is the peptide, basically. It's a 15 amino acid peptide, and what it does is it actually is attached to the calcium phosphate matrix, and when it's attached to the matrix, it increases the number of binding sites for the cells. So they are attracted to, bind to, and then begin activation and proliferation and formation of bone. So if we look at the activate or attracting, what we do is we actually increase the amount of cells that are actually drawn into it and attaching directly to the matrix itself. In some of the studies, we actually found that when it attaches, it actually also sets out pseudopodia and changes the endoskeletal structure of the cell itself. Once it begins sending out the extensions, it actually has a haptotactic effect where it begins to migrate along the surface of the material, increasing bone formation, which is not really the same as chemotaxis, which is something that does occur with, say, BMP and some other products. We can see the expression of matrix was much, much higher. We could actually see that the cells actually bound to it at a higher level, and also the amount of surface area that was covered by cells was also significantly higher because of the attachment factor. Activation, again, we went through the integrin molecule interactions, and some of the downstream effects that were associated with mRNA expression and so forth were that we were actually able to see, again, a larger pseudopodia and endoskeletal structure changes within the cell, and at the same time, we're able to measure gene activation. We can actually see an increase in alkaline phosphatase, significantly greater than material that did not have this added to it. And also the BMP2, we actually looked at BMP7 as well, but actually we actually demonstrated significantly higher levels of BMP2, both expression within the cell and expression into the surrounding matrix. It also increases bone formation. You can see it with these mineralized inclusions away from the graft into exactly the area of implantation. And also, early mineralization was due to the expression of annexin 2, which is a very important integrin that is utilized with cells for attachment and stimulation of bone formation. That's one of the ones that we were actually able to measure more closely. We actually have a couple of ongoing studies to try and understand that and ligand interactions as well. We have much more early mineralization because of these interactions. And the mechanism of action is, it's an attach, attract, and activate. It's not osteoinductive. There is a secondary osteoinductive component to it because, as you saw, it actually upregulates the internal production of BMPs, expression into the environment. But it itself is not an osteoinductive product. And it's not a morphogen. It does not cause one cell, or say a mesenchymal stem cell, to morph into, say, an osteoblast. It does not have that same activity that a BMP does. It's not a chemotactic product because it's bound terminally to the calcium phosphate matrix instead of having a diffusion gradient to allow for cells to come in through an inflammatory response. It doesn't work that way. There has been no ectopic bone formation that's been identified in our early data, even subsequent data after the approval of the product, plus subsequent data through outside the United States as well. And it does not act away from the implanted site. So if it does migrate out, it's usually resorbed by the body without forming bone in an ectopic fashion. The technology was started in dentistry and oromaxillofacial surgery back in the 1990s. So we've been on spine here in the United States since November 2015. It's been available outside the United States since 2007. And we have about 40,000 cases that have been completed and quite a bit of data to follow up as well. It's PMA approved here in the United States for an anterior cervical and discectomy fusions at this point. And we actually are starting another study, and Dr. Chapman and the group here will be involved as one of our sites for a TILA study. So that's something we're going to look to try and increase our footprint and indications. The procedure that we actually studied on was an anterior cervical discectomy and fusion, 319 patient enrolled in 24 sites, one-to-one randomization. The primary endpoints, of course, were fusion. We looked at NDI, VAS, pain, and osmometry, and also the SF-36. And we also follow them all the way up to 6 years. We have some of the 5-year data coming out now. We'll have 6-year data coming out fairly soon. And we've been able to demonstrate continued success of the product through that time period. The study itself was, you can see it, 1 and 2 year time periods. We looked at diffusion success, NDI, neurologic, and then safety. But when we looked at the composite scores of all of the parameters, it actually outperformed Autograft. But again, we set this up as a non-inferiority study, so we're at least equivalent to Autograft, according to all FDA evaluations of the product. It has superior clinical safety profile with long, broad experience compared to other products that have gone through exactly the same type of a study. It's the only FDA bone graft right now available for cervical fusions, and it's a storage, of course, at room temperature, and there's no preparation necessary. Just open the package and go ahead and utilize it. There's quite a bit of supportive evidence for a number of other products that are out there. Autograft, yes, it works. We've known it for many, many years. Infuse, absolutely been proven to be equivalent to Autograft, and now the I-Factor product also has that same level of efficacy. We look at some supportable evidence in other areas. Allograft in some low-demand applications has been proven very, very efficacious. Some other synthetics, VTOS being one, Helos another one, when bone marrow is added to them, actually form bone quite well as well. But, the evidence really doesn't seem to be there for most of whatever else is on the product, other products that are on the market, synthetics, the stem cell-based products. And the DBMs, while it's been variable as far as success rates, as a standalone product, really has never borne to be equivalent to Autograft at this point. So I talk kind of fast. I apologize for that. But thank you very much for your attention and any questions.